Everyone. So today we're going to continue our molecular biology unit, and we are going to start lesson number 6, which is translation. This is what your note taker looks like. It is 2 pages. So put on your thinking caps and let's begin. Okay. All right, so once again, you're probably tired of hearing me say this, but let's review the central dogma of biology. DNA can make more copies of itself, that's known as replication. DNA can make RNA, known as transcription, and RNA can make proteins, that's known as translation. This is the central dogma of biology. And we know that DNA is the one that provides all of the instructions for assembling the amino acids, which in turn is what forms these proteins. And something interesting to keep in mind is that the code for specific amino acids, so the amino acid or the building blocks that build up proteins, is virtually the same for all life forms, which talks about our evolutionary history a little bit. So we want to remember that there are 3 types of RNA, but today we're going to look at their role in translation. So we're going to define a little bit in more detail what their individual functions are. So we're going to start with mRNA, and mRNA is what carries the genetic information from DNA to the ribosome to make proteins. And mRNA contains codons. So codons are sections within the RNA strand made up of 3 nucleotide pairs. Oops, sorry there. Okay. tRNA, we see right here, tRNA, it brings the amino acid. So this little blue structure here, this is the amino acid. And tRNA brings the amino acids to the ribosomes to help build proteins. And tRNA has what is known as an anticodon, which is the corresponding bases or nucleotide bases that will match up with the codon on mRNA. And then rRNA. rRNA is actually kind of an incredible type of molecule because we know that not only does it help with translation, but it also has enzymatic capabilities. That means that it acts as an enzyme. And it combines with proteins to form ribosomes. So the ribosome that is actually used for translation contains our RNA and ribosomal RNA. And some of the functions of ribosomal RNA are still not known. All right, so a reminder, here we see the 3 main processes in the central dogma in biology. The first process is DNA replication, and that happens in the nucleus. The second process is transcription. That also happens in the nucleus when DNA makes RNA. And the third process that we're focusing on today is translation. And this happens on the ribosome, which is free floating in the cytoplasm of the cell. Okay, and that's what we're going to be focusing on today. So translation, what is it? Well, this is a process where RNA makes proteins using a ribosome. Where does it happen? It happens in the ribosome. When does it happen? It happens during interphase. And it uses start codons and stop codons to tell the RNA where to begin and where to end building a protein. So we're going to learn in just a moment that proteins in your body have lots of different functions, right? But depending on the cell and depending on the organ, you're going to need to make a particular protein to carry out a particular life function. That protein has a particular size. So RNA needs to know how big that protein is going to be. And RNA also needs to know when that protein needs to be made. So these codons, or these sequences along the RNA, are important in signaling the beginning of protein synthesis. But also the polypeptide cannot just continue growing indefinitely. There needs to be a stop sequence or a stop codon on mRNA that tells the protein sequencing system to stop producing that protein. Okay? So what is the codon-anticodon relationship? So write down the definition of a codon. A codon are 3 nucleotides in mRNA. So we find the codon here on mRNA. Okay, so 1, 2, 3 nucleotides are 1, 2, 3 bases. Each codon will code for a single amino acid. So for example, this codon of GAG will code for a particular amino acid that's here on the tRNA. Oops. Now the anticodon, these are 3 nucleotides, but the 3 nucleotides are found on tRNA. So notice that tRNA kind of looks like a T, right? If you think about it. So this is tRNA here. It's transfer RNA. It's called transfer RNA because it's transferring an amino acid that it carries here on this end. And mRNA, notice that it also looks different. It's more linear, right? And it's called messenger RNA because it brings the message from the nucleus, from DNA. So the relationship is that the codon on mRNA is going to molecularly bind with the anticodon on tRNA. Notice how the bases are complementary. G with C, A with U, and G with C. Okay. All right, so this is what the translation process looks like. Let me open my pen here. So here we have mRNA, right? And we see that mRNA is pretty much a linear sequence here. And mRNA is going to have a start sequence, okay, at some point. And tRNA is going to come in, and tRNA, which is right here, is going to bind its anticodon to the codon on mRNA. So notice that there's going to be a chemical reaction here, a binding of the anticodon of tRNA to the binding of the codon on mRNA. And tRNA is going to carry an amino acid that is complementary to the codon on mRNA. Okay? And then this amino acid is going to go through some chemicals processes here, and it's going to bind with a peptide bond to these other amino acids. Okay? So we see that the polypeptide chain, or the chain of amino acids, or the protein, is growing. Right? It's getting bigger and bigger and bigger as amino acids are added to that protein chain or that polypeptide chain. All right, let's continue then. Oops. All right, so we have a codon table, which is the code for specific amino acids, and it is virtually the same for all life forms. So you might see a codon table that actually looks like a table, like this one, okay? Or you might see a codon table that it's not in the form of a table, but it's actually in the form of a wheel, like this one. So you're going to need to be able to interpret both the wheel and the table. So notice that there are approximately 20 common amino acids to build proteins. So all these weird little names that you see here, PH, E, L, EU, all of these are amino acids. So this is the amino acid known as phenylalanine or leucine or isoleucine or methionine or valine. So all these are amino acids. And notice that each amino acid corresponds to a particular codon, like UCU or CAA. So for example, if you want to know which amino acid codes for proline, which codon codes for proline, it could be any of these codons, CCU, CCC, CCA, or CCG, okay? So the start codon is always going to be the same. So AUG is the start codon. So let's find it right here. AUG is the start codon, is going to signal the beginning of protein synthesis, and AUG codes for the amino acid known as methionine. Okay? And then there's going to be 3 stop codons, which stops translation or protein synthesis, and these stop codons on mRNA are always going to be the same as well. They are known as UGA, UAG, and UAA. Okay, so here you see them, stop, stop, stop. Okay? Okay. So actually, before we move on to this, let's look at your note taker because you have I give you a list of 4 different codons, and I want you to find the corresponding amino acid. So I give you the codon AUG, and I ask you, what is the corresponding amino acid? So we know that AUG is right here, okay? And the way you read it is you find the first letter, A, the second letter, U, so that means you're here in this column, and the third letter, G. Okay, so AUG codes for, they all meet up here in the same place for methionine. Now the wheel, you're going to read it from the inside out, okay, from the inside out. So how do you read the codon wheel? You're going to find AUG the same way. Here's A, here's U, and here is, can't see very well, there it is, and here's G, AUG. And again, it codes for methionine, right? So it's going to give you the same information in the wheel and the table, okay? The next codon I want you to code for is UCC. So see if you can do this one using the table. So here's U, the first letter. Here is C, the second letter. So that means that U and C meet up right here in this box, okay? And then the third letter is C, and that's going to overlap right here, UCC, and that codes for the amino acid serine. And you can see that UCC, again, UCC codes for serine, just the same way, okay? The next one, try it on your own. What amino acid is CAU coding for? So try to do that on your own. What is CAU coding for? You got it? Yep, it's the amino acid histone. Okay, and here it is again, CAU, and there it is. Okay, and the last one is UGA. And we know that UGA is actually a stop codon. So it codes for the end of protein synthesis. All right. The next slide just has you looking at the different parts of translation. And I want you to take some notes regarding the process. So label the parts and summarize the process. So let's start by labeling the parts here. So here we see number 1 is pointing to the actual amino acid. So this is the amino acid that is helping the protein chain grow, right? So as tRNA comes in and adds amino acids, this chain of amino acids, or this polypeptide chain, is going to grow, forming a very, very long protein. Remember that a protein is also known as a polypeptide or a chain of amino acids. So here we see that the amino acids are held together by a peptide bond. So you were wondering why a protein is also called a polypeptide. Well, that's because the amino acids are held together by peptide bonds. All right, number 3 is showing you this structure right here, and this is the larger subunit of the ribosome. So if you recall when we studied organelles, we know that ribosomes are made up of small and large subunits. So this is the large subunit of the ribosome. Okay. Number 4 is pointing to this whole structure right here, and this is your other type of RNA known as tRNA, or transfer RNA, carrying the amino acid. We know that tRNA carries the amino acid on one end, and it carries the anticodon, or the sequence of 3 bases on the other end. So tRNA carries the anticodon on the other end. Here we see for number 7 that mRNA, which is the linear form of RNA, is translocated along the ribosome. So this is your mRNA. For number 6, we are looking at the small subunit of the ribosome. So this is the small part of the ribosome, or the small subunit of the ribosome. And number 5 is actually just focusing on the 1, 2, 3 nucleotide bases on the mRNA, and we know that that is the codon. Okay, so these are all the players in translation. So how do we summarize the process? Well, first you need mRNA to come in from where? Where is mRNA coming from? It's coming from the nucleus carrying the genetic code, and mRNA translocates or attaches itself to what? To the ribosome. And to be even more specific, it's kind of sandwiched in between the large subunit and the small subunit of the ribosome. Okay, so what happens after RNA attaches to the ribosome? Then tRNA comes in and it attaches its codon, it attaches its anticodon to the codon on mRNA. So tRNA anticodon attaches to the mRNA codon. And remember that the tRNA, as it attaches, it's also bringing in the amino acid. So there's different regions here within the mRNA where the tRNA kind of attaches itself, and then the amino acids kind of link up, and then the tRNA then leaves. So there's 3 different processing sites here that you're going to learn more detail when you take AP or IB Bio. But after tRNA kind of hands over its amino acid, the amino acid chain begins to grow. So amino acids then connect or bond. Better to use the word bond here. So amino acids bond forming a what? What happens when you have a chain of amino acids? Forming a protein or a polypeptide, which is a chain of amino acids held together by peptide bonds. Okay, so that's how this protein is just going to continue growing and growing and growing. Okay. Okay, let's continue then. All right, so then what happens after the protein gets made during protein synthesis? Well, it's not ready to be a functional protein yet. It needs to get modified, and that's what we know as protein modification. And there's different levels of structures and modification of proteins, and depending on the structure of the protein, that protein is going to have a particular function. So we know that in biology, form fits function is a very important idea to remember. So these proteins aren't all going to have the same function. Some of these proteins are going to be fibrous in nature, so they're going to be structural proteins. And then some of these proteins are going to be globular in nature, which means that they have an enzymatic role. They're going to catalyze some type of reaction here. So the protein gets modified and you get different structures. So you have a primary, secondary, tertiary, and quaternary structure that you don't need to know in whole detail what their functions are. Just note that there are 4 different levels of protein structures here. Okay. And once you have the different proteins, whether you have structural proteins or globular proteins, these proteins are going to have a different function. So here we see some of the different functions of proteins. Like proteins can form your hair and your fingernails, or they can form major components of feathers, wool, claws, scales, horns, hooves. We have proteins forming your muscles. We have proteins acting as cellular messengers. We have receptor proteins that transmit signals. We have proteins acting as antibodies that are part of your immune system and help you fight off infections caused by bacteria or viruses. Oops. We have proteins in your blood, for example. Hemoglobin is found in your red blood cells, and it helps transport oxygen in your blood. We have proteins in your brain and nerves acting as ion channels that control brain signal. We have proteins that act as enzymes in your saliva to break down the food that you eat, in your stomach, in your small intestine, all helping in food digestion. And then you have proteins acting as cellular construction workers, such as molecular machines that do your cells' heavy work, like copying genes during cell division or making new proteins. So the functions of proteins are definitely indeed. We could not be able to live without our proteins, and that's why we have thousands and thousands of proteins, all with different functions. Now, one last note to remember, and we saw this in our last lesson, that not all genes are able to form proteins, right? So not all genes within the DNA are structural genes that are able to be translated into proteins. Remember that gene expression is an important component of understanding how proteins get made, because there are some genes in DNA that are regulatory genes and do not get expressed as proteins. Only those structural genes are expressed as proteins. And also there are some genes that we don't even know their function yet, right? And do not get expressed at all, or get expressed in weird situations. Something that you'll learn later on in higher level biology is that sometimes the environment can affect also gene expression by how DNA gets methylated. But that's something that we will explore later on in higher level topics. All right, that's it for today. I hope you enjoyed your lesson on translation.